So the first paper is this Advanced Physiological Sciences article, and I'm not quite sure what year this was because they don't put it in the citation. But this is volume 16, Sensory Functions. This is Anatomical Basis for Divergence, Convergence, and Integration in the Cerebral Cortex. So as I mentioned before, he was a biocyberneticist. There's a biocybernetics wing of the Max Planck Institute. So they have this, they still actually have biocybernetics as an area of specialty. And I guess in this case, what they're doing is looking at mainly biological systems like the brain in this sort of cybernetics way. And I don't know what cybernetics means now, but this is what Valentino Braitenberg was thinking about. So what his concern was here was looking at the anatomical um, aspects of cerebral cortex, and then looking at some of these, the electrophysiology of it and the anatomy and how that comes together. So he talks about cortical electrophysiologists, and he says they're beginning to generate functional models which must eventually be fitted into the known structures of the cortex. So this was many years ago now. So I'm not sure if this is still true, but I guess we know have functional models now. But again, we have to fit this into the known structure of the cortex, and that can always be challenging. Quite independently from this ancillary role of histology, the structure of the cerebral cortex is impressive enough to invite speculation on its own. Particularly, the view of the cortical computer, derived from the electrophysiology of sensory motor areas, is quite different from that which the global view of the cortical anatomy inspires. So there's this idea of the cortical computer and how this is based on the electrophysiology of certain areas of cortex, and then there's this global view of cortical anatomy. These two views will eventually converge. As a counterweight to the prevailing trend, I shall collect here some obvious facts from cortical anatomy, which are frequently overlooked, although they might significantly contribute to our ideas on the working of the cortex as a whole. So he was struggling with this idea that there's this, there are these areas of cortex which we study quite intensely. We have computational models of those, and then we have cortex in general and what that is. So this is again kind of thinking about the neuroanatomy, but also thinking about the function, and then kind of putting that together. So he kind of goes through the macroscopic anatomy. He goes through the description of the synapses. This is all descriptive, so this is not necessarily, it just tells you what's there. And then talking about some of the classification of synapses, and then the density of neurons, the types of neurons, relative number of the three types, axonal density. So it's like this kind of laundry list of what he's seeing in this model. This is, this is the albino house mouse. So this is being compared to human cortex, but focusing on the albino house mouse. So you go through all of this description, divergence and convergence. So this is interesting. If this is a learning machine, it is important that it be free to learn whatever patterns are presented. There must be the possibility of a connection from every neuron to as many as possible other neurons. If a pyramidal cell has 5,000 synaptic contacts, with how many different cells is it connected? So this is where we have one cell being connected to every other cell, having this fully connected architecture. And so there are two lines of arguments to suggest the answer is with almost as many, and this is cited in Braitenberg, 1978. First, we consider that the cross-section of the dendritic tree of the neuron and derive a geometric measure of the probability of a fiber entering its confines and hitting it once, twice, etc. The probability is very small for 2 or more hits. So I think this is a picture of this where you have this dendritic tree and you look at the architecture of the tree and see how many times it sort of intersects, and you're getting a measure of the sort of full connectivity of the cortex. So this is a cross-section of the basal dendritic tree of a pyramidal cell, as seen by a straight fiber entering its confines. So this just shows this. And then there's this probabilistic estimation that's made. What are the surface area of this region and the area of the circumscribed circle? The probability of hitting it twice is proportional to the small hatched area here. So these cross twice, and you see this here. Second, we may picture a neuron as a cloud of postsynaptic sites, intermingled with many thousands of clouds of postsynaptic sites of other neurons. If a fiber entering that region picks its contacts at random, we may calculate the probability of it hitting that neuron once or many more times by the binomial distribution. Here again, it turns out that the probability of multiple hits is quite small. So he's working out this problem by doing sort of a topological model and figuring out how, if you just generate connections, how often do they cross each other, and you can calculate that probability. And then doing a sort of a probability density function approach where you just assume just a cloud of interactions. And in both cases, it turns out that this is quite a small probability of happening. So what does this mean? This means ultimately we derive the picture of the cortex as a device which reshuffles the incoming information in as many as possible ways, perhaps in order to discover correlated activity and to incorporate this knowledge in the form of what he calls cell assemblies, which of course are a thing that he proposes in 1978, cell assemblies in the cerebral cortex. And then, we have more modern work that kind of expounds on that. So that's the first paper. The second paper is this brain size and number of neurons, an exercise in synthetic neuroanatomy. This is more recent. This is from 2001. And so he kind of gives these different models of sort of invariances in comparative neuroanatomy. So let's go through the abstract. Certain remarkable invariances have long been known in comparative neuroanatomy, such as the proportionality between neuronal density and the inverse of the cubic root of brain volume, or that between the square root of brain weight and the cubic root of body weight. Very likely, these quantitative relations reflect some general principles of the architecture of neuronal networks. So we have these quantitative aspects. We have brain volume, brain weight, body weight. So this is the sort of allometric scaling that you'll see in biological systems. Brain growth is, a brain can grow, but it's usually with respect to the body. So you get bigger brains in bigger animals, smaller brains in smaller animals. And then that scaling can change itself. That scaling function can change its slope based on the expansion of cortical regions, or sometimes in birds, it's the telencephalon. And so you get this sort of scaled relationship, and it gives us these quantitative measures that we can get a sense of the architecture of neuronal networks. You also see this in the hippocampus of different organisms, especially if they engage in a lot of foraging versus not. Under the assumption that most of the brain volume is due to fibers, we propose 4 abstract models. So the first is constant fiber length per neuron. 2 is fiber lengths that are proportional to brain diameter. 3 is a complete set of connections between all neurons. And 4 is complete set of connections between compartments, each containing the square root of the total number of neurons. So these are 4 abstract models. These are just assuming different things about the brain volume and what's inside there. So we kind of think that most of the brain volume is due to fibers and connections packed in there. We also, can assume maybe a constant fiber length. So if we assume a constant fiber length, and we assume that most of the volume is due to fibers, we can then calculate the number of neurons. We can also assume the fiber length is proportionate to brain diameter. We can assume a complete set of connections between all neurons, which gives us maybe a number for the number of connections and then the number of neurons. And then 4 is a complete set of connections between compartments, each containing the square root of the total number of neurons. So this is where we have modularity. We have these modules that are connected, selective with very few connections, and then each module is fully connected within. And then so talks about the models in terms of what they correspond to. Model 1 conforms well with the cerebellar cortex. Model 2 yields the observed comparative invariances between the number of neurons and brain size. And this is, of course, this allometric scaling idea. Model 3 is totally unrealistic. So that is our complete set of connections between all neurons. So that's actually totally unrealistic. And then model 4 is compatible with the volume of the hemisphere white substance and different from mammalian species. So what this paper is suggesting, kind of is maybe in contrast to the other paper, but also maybe supporting the other paper, is that we can think about the brain or the volume of cortex in terms of how it's connected. We can work backwards, find the number of neurons, find the connectivity patterns, and then, make certain assumptions about those connectivity patterns and see how well that model fits the data. So, no, we don't have fully connected neural networks. We actually have maybe this modular network where there's maybe local full connectivity and then selective global connectivity. And that would fit what we see in mammalian species. So he goes through, a lot about this sort of set of assumptions, how these are set up. We have these 4 models and talking about real brains. Unashamedly unrealistic as our schemes of 1 to 4 may seem, they do yield some values that are remarkably close to measurements of real brains. And so we're going to go through some of these comparisons. And so, we know from experiments that allometric scaling is a 2/3 power law. The number of neurons and the 2/3 power of brain weight is something that was established experimentally back in the 1950s and 1960s. So this is where we have the allometric scaling for the number of neurons versus brain weight in grams. So the weight of the meat or the weight of the sort of the tissue versus the number of neurons within that volume of tissue. So you can see that, monkey, cat, rat, and mouse all fall along this slope. And then humans are out here as an outlier. And the reason humans are out as an outlier is because in a lot of primates, you have this expansion. In great apes, you have this expansion of the brain. And in humans, you have the greatest expansion of brain tissue. And so you have this outlier out here. Okay, so then, this just talks about thinking about this approach of what he calls synthetic neuroanatomy, where we're using simulations, we're using mathematical models to model a lot of this stuff. And so I think it's a useful approach. It's definitely something that we've picked up on in our group. And this is a slide I made up of these 4 models of the brain. So in black is from this paper we just talked about. And then in red is sort of the way that this is dealt with in the developmental neurosimulation paradigm. And if you're interested in more about this, we have a series of papers on our website that have been published in various places that go over some of these concepts. And so developmental neurosimulation kind of addresses all four of these models in different ways. And so if you think about like a vehicle, because he's talking about real neuroanatomy, but eventually he gets to this sort of model of vehicles and, thinking about the vehicle versus the actual brain. We'll talk about vehicles here, but he's talking about sort of real brains. What we're interested in is using vehicles as a means to get to simulated brains or developmentally relevant brains. So then the first model then is brains of uniform density. And this is where we have a compartment which contains a brain. The density is uniform across that. The volume is sort of, there's a volume and a density of content. So there's this little container here, this gray region that has a certain density and within that density we have neurons and axons, dendrites, so we have the connections and the neurons. And in developmental neurosimulation, this is a size of a network that's growing. So in developmental neurosimulation, our network is constantly growing within a volume, or the volume is expanding, and this expands the size of our network. And so we kind of approach this in a little bit different way. Remember from our last discussion, Braitenberg kind of downplayed the significance of development, or at least he didn't talk about development in his work. In our case, we're explicitly interested in development. So we're interested in the size of this neural network and this network of connections. And for a uniform density, we probably have a fully connected network. And so we characterize our matrix of network connections as a determinant, which is here, A. And so what is the size of A? And so in developmental neurosimulation, you have this determinant that gets recalculated over time as the network grows. And so this grows in size. And this can be also stated in terms of density. I've not done this here, where you have a restricted space in which you can put more and more neurons. So the size, I should also probably put the density of A as well, but I'll do that later. The second part is this network here in this vehicle from sensor to effector, which we can characterize as Wij. And this is where the length of each of these connections is proportional to the diameter of the brain. So as Wij grows, as the body grows, this is in Braitenberg's work, this is brain-body scaling. In developmental neurosimulation, this is also brain-body scaling. And in this case, we don't have just a compartment of a brain, we have this mapping from sensor to effector, which as this distance between the sensor and effectors grow, or the relationship between them changes, we get a different length of our connections. So our connections are, we have things that are connected. So we have sensors connected to interneurons, connected to effectors. We can describe that with Wij, but we also get more connections. We also get a longer distance between the sensor, the interneuron and effector, and so forth. So we can characterize Wij both in terms of growth, in terms of number of units, but also in terms of the length of these connections. And of course, biologically speaking, the length of the connection is important because the longer the connection, the more conduction delay you get, and the more leakage you can get unless you have myelinated axons. So these are, things to consider. I don't know if we consider this in a synthetic model, but definitely in biology. Then models 3 and 4 are the first. Number 3 is a complete set of connections between neurons, which is just basically this mass of connections without any modularity. And then 4 is where you have this fully connected end to the half compartments. And these are modules with limited connections, but internally with complete connectivity. And so we can bridge those two models actually in the developmental neurosimulation paradigm by saying that a selective set of connections between neurons involves n to the half compartments. And so we can actually have these kind of compartments play the role of interneurons or other modules in an agent brain. Whether those are functional or not is, we haven't really gotten to the point where we would test those kinds of hypotheses. In any case, so let's go on to the next paper here. And this is from 1974. So he was doing his work in this, from the 50s to 2001. So this is like a long period of time where he's working. But this is from 1974. And again, he was doing a lot of neuroanatomy. So some of this is just really cut and dry neuroanatomy. It doesn't think about too much about the simulation aspects or the modeling aspects. But in the last paper, as we saw, there is this very strong simulation aspect, biocybernetic aspect. So summary of this paper, he's working in the visual. So in the first visual ganglion of the fly, Musca domestica, there are many similar visual channels or cartridges connected to each other by various systems of fibers, the most regular of which consist of the collateral branches of the L4 neuron, which is contained in each cartridge. The three collaterals of L4 run to three different cartridges, one of which is the parent cartridge of the neuron, the other two being neighbors of that cartridge in two directions on a hexagonal array. Within each cartridge, there are thus the endings of three collaterals from three different L4 neurons. These make close physical contact, and serial sectioning shows that each collateral is presynaptic to the other two. It follows that there are reciprocal synapses between any pair of these collaterals. And then the network of relationships thus set up is a simplest scheme of coupling in a hexagonal array. So he shows images of this. So he works in this area where he's thinking about flies and their visual ganglion. So in dipterians, the visual field is resolved in discrete points, each corresponding to the central axis of one ommatidium. So the fly visual system is different from the mammalian visual system, in that they have these small ommatidia that form discrete points or dots in a visual array. So, we have sort of this, call it a camera-like eye, which has like spatial resolution, more continuous spatial resolution. We have, we can detect shapes and blobs and things like that. In the fly visual system, we have these dots, and you can present things in terms of dots, and they find patterns between dots. And so this divides things up into these subunits, and there are thousands of subunits in the eye. They function much differently than our retina. So this is, kind of the basis for a different type of visual system, and then looking at how that's organized. So this is the arrangement of L4 collaterals between the L4 main fiber, which is the black dot, and the cartridges, the white circle. So you have this sort of organization. It's a different type of organization than the mammalian visual system. And I think this is important because a lot of what we do in pattern recognition in our studies of visual perception, say using a neural network, is to assume this sort of model that is presumptive of mammalian cortex. And this is not mammalian cortex. This is a very different way of organizing a visual system. And so this is just showing how these systems are organized, how they're wired up, and then how they function. So this is an electron micrograph of a synapse. So this is an electron micrograph where you see the different structures. They're pretty well resolved. And he shows he's labeled L3, L4, R6. And the receptor cell axon is R6. The neurons are L3 and L4. And you can see the connections here. L1 and L2 are over here. So he gets through the neuroanatomy, talks about the connectivity, He has this diagram of reciprocal contacts between L3, L4 end feet. And so this just shows what the cartridges look like. So remember, this is all neuroanatomy, and one of the bases for Braitenberg vehicles was to say, okay, there's this very highly specific anatomical relationship between sensor and effector. If we look at an insect, we can see that there are these structures in the sensor. We have these structures in between the sensor and effector, which is kind of the brain wiring, and then the effector, which has its own structure. And so what he's doing is working out all this. The idea is that if you work out the anatomy enough, you can get this very precise model that can produce all sorts of behaviors, which is what he was interested in the book Vehicles. And so this just really kind of describes this sort of system. Our findings indicate that reciprocal synapses, as described in Rawl et al. 1966 in the olfactory bulb of the rabbit, may be a more widespread feature of nervous systems than has hitherto been supposed. So his argument also is that some of these aspects of neuroanatomy, of wiring, are universal across different nervous systems. So we should lean into those universal aspects. So yeah, he talks quite a bit here about symmetry and about some of these synaptic relationships and how they might be, really kind of, if we can characterize them well enough, we should be able to characterize the behavior they produce. He also has this other paper from 1974, Thoughts on Cerebral Cortex, talking about his thoughts on cortex. So this is kind of where he thinks about it as an input-output system, much in the spirit of cybernetics. So input-output means you have a sensory input, you have some sort of output, and then that gives you behavior. So I'll just read the abstract here. So the cortex is often described as a network processing information in the direction from sensory to motor areas. So going from sensors to effectors, the motor effectors, the sensors that are bringing in stuff from the environment. That requires an input of sensory information, an output of motor behavior, and something in between. So this is kind of the assumption. However, the structure of the cortex is asymmetrical only in the vertical direction, suggesting an input-output transformation between layers rather than between areas. So if you think about the brain as, having these modules or having these layers, the asymmetry is only in the vertical direction. This operation must be a very generally applicable one, since the plan of the cortex is basically the same everywhere. So in an attempt to understand it, a skeleton cortex of only pyramidal neurons is considered. They are characterized by a double dendritic expansion, an apical one in which the first layer, which is considered as the input layer, and a basal one which receives excitation from the axon collaterals of other pyramidal cells. So when he talks about layers, he's talking about the layers of cortex going from 1 down to 6 and kind of coming back up. That's what he's kind of getting at with this. If pyramidal cells learn, perhaps by growing dendritic spines to respond to frequent constellations of activity in their afferents, each will learn a property of the input through its apical dendrites, provided it was preceded by other properties sensed by neighboring pyramidal cells, which influence it through basal dendrites. So in these layers of cortex, you have a lot of feedback, but you also have this functional specificity for each layer. Thus, the pyramidal cells will code its input in terms of properties, which have a tendency to follow each other. This will be a coding which reflects the causal structure of the world. Various uses of a network embodying the conditional probabilities of events in the input is described, including the recognition of familiar sequences and prediction. The local variation of fiber patterns in the cerebral cortex, described as myeloarchitectures, is interpreted as a macroscopical expression of the different statistics of the set of conditional probabilities linking events represented by individual pyramidal cells in different areas and in different functional contexts. So this is going through his thoughts about the sort of the vertical asymmetries of cerebral cortex, much more than between regions, and how that plays. So he's really kind of mapping out this sort of theory of cortex. And this is not something that the Numenta people talk about. They focus a lot on Vernon Mountcastle's work on discovering these kind of layers of cortex and their kind of relationships between one another, thinking about things in these cortical columns, which is just a stack of these different layers of cortex arranged in units that do things like process images or um, process sound, and kind of going through each layer, doing different aspects of the processing. And so, he gives us conventional view of cortex as sort of these modules that are distinct sensory areas. So you have connections between, say, sensory motor and motor. You might have visual and auditory, and you have to move between the regions. And so this is kind of these regions of cortex, and then you have the symmetry of the cortex, which is this layered cortex. So he says the anatomical picture does not bear out this sort of model of distinct regions. If I'm given a small cube excised from a logical network showing the same asymmetrical pattern on 4 of its 6 sides, and an isotropic organization on the other 2, I shall hardly suppose that the network processes information in the direction indicated by the arrow A parallel to the layers of the asymmetrical organization in B. So as you move across the surface of the cortex, that's a different thing than moving up and down the cortex. That's kind of the point he's making. Okay, and so just thinking about the anatomy of the cortex gives us sort of, you have to reinterpret that in this differential view. So, he's talking about this in 1974, and actually he's modeling the cortex using the skeleton cortex, where he's kind of thinking about it, just stripping down everything in the cortex to these pyramidal cells. And again, this is an early version of computer simulation, or, thinking about this in terms of a computer simulation. And so this is a basic network of pyramidal neurons or pi neurons showing the afferent fibers from the first layer, apical dendrites, basal dendrites, and so forth. So these are the apical dendrites, these are the basal dendrites. So this is it's a pretty good paper. This actually reflects a little bit on what pyramidal neurons represent. So each pyramidal neuron is connected in its own special way to a particular set of fibers in the input layer through the branches of its apical dendrite. We may say that each pi neuron represents a property of the input in the sense of Klein's 1956 representation of events in nerve nets, namely in the sense that the pi neuron becomes activated when an event has occurred in the input that is the property of which the pi neuron stands for. We disregard, for the moment, the important question of how much synaptic influence under the peripheral portion of the apical dendrite will ultimately arrive at the initial segment of the axon. But we can model this anyways. So he builds this nice model and uses pi neurons as memory elements as well. He thinks about this in terms of the engram and in terms of different types of associative learning and how that relates to modifiable synapses. So he's really thinking of this at the anatomical layer, but he has interest in, traditional cognitive questions like memory, associative learning, and so forth, sensory motor learning. And so the assumption of this kind of memory in the pi neurons throws new light on the question of what they represent. Having learned in the fashion described, they represent properties of the input which have a tendency to occur in a certain sequence. So he's thinking about sequential learning or sequential information. Or if emphasis is not as much on the temporal order of events as on the fact that occurrence of one event raises the probability of the occurrence of another, such as in Hebbian learning. We might say that pi neurons represent those properties of the input in terms of which the causal structure of the world represented becomes evident. If a connection axon collateral-based dendrite has been established as a synapse because it has frequently contributed to the state of excitation of the neuron. It follows that the synapse is a record of the conditional probability, linking the activity of the presynaptic neuron with that of the postsynaptic one. So in terms of the outside world, that synapse stands for a causal relation between the events represented by the two neurons. So he's not just suggesting there's associative learning, but that there's this causal representation. So, that's something that may be controversial even today to think about this in terms of a causal relation, because there are a lot of feedbacks as well. And, that's something that I don't know if he modeled that, but that's something in the brain that you often have. And knowing, like, there's this whole area called credit assignment where you're trying to figure out, like, what neuron is contributing or what set of neurons is contributing to what behavior. This last paper are two views of the cerebral cortex. This is from Brain Theory, 1986. And this kind of talks about cerebral cortex again. And he talks about reciprocal positive feedback. And so he makes this comment here. Cortical anatomists and physiologists learn to shape their findings so as to make them acceptable to the theoreticians. The resulting situation of reciprocal positive feedback has some stable solutions. So there are a couple, well, let's see, how many are there? There's 5 solutions here. The first is random networks with and without learning. Lashley's philosophy is of this category, as is Hebb's theory of cell assemblies. And so Hebb also had this theory of cell assemblies. Rosenblatt's perceptrons is also a descendant. 2, the circuit diagram is in the spirit of radio engineering. The amplifier entered neurophysiology from communication engineering. With it came various ideas. So this is kind of focusing on this aspect of cybernetics as a circuit diagram. 3, the digital computer and a logical theory of nerve nets. This was soon recognized as a misleading analogy that the digital computer has, at any rate among all models of cortical function, the unique distinction of being a very useful machine. And the theory formulated by McCullough and Pitts, made more palatable by Klein, lent the brain a flareable mightiness for which was gratefully recognized by many. 4, the idea of projection derived from optics. So this is where you get these maps of the body surface or some sensory space repeated many times in cortex or in subcortical structures. So again, projectionist research is still flourishing, but we don't really understand the details. I think we're better off now than we were then, but still, this is sort of derived from optics, where you're projecting things in different ways through a lens. In this case, the lens is through the nervous system and processing of the sensory organs. Number 5, the idea of localization of function, derived from dualist philosophy. If you are utterly convinced that the connection between mind and brain escapes us as a matter of principle, all you can do is find out what part of the brain is related to what faculty of the mind, without asking any further questions about the nature of the connection. This leads to very useful partitioning of the brain and perhaps less useful dissection of the psyche on anatomical grounds. So he kind of goes through these points, and then in view of these findings, it would seem difficult to sustain the essentially random network philosophy, which has grown dear to our hearts on the basis of many anatomical facts. However, I will show that it is possible to make the 2 points of view quite compatible with one another. So he goes through this talking about the neural pill in the mouse cortex. So he says that all the essential features of cerebral cortex can be found in the mouse, except that in humans it's scaled up by a factor of 1,000 in terms of size. It is a task requiring some experience to tell a histological section of a mouse cortex from a human one. Before we approach the problem of the shape of neurons in the cortex, we take a more global view and collect some quantitative data. So that's what he's doing here. Making some measurements, the density of neurons, distinctions between synapse types, axonal density, and then the axonal trees of various neuron types, dendrites, the cross section of a dendritic tree. So there are a lot of measurements you can use from the neuroanatomy. In principle, these figures provide the basis for a connectivity matrix of the cortical neurons, but of course the form factors reflecting the shape of dendritic and axonal ramifications are also involved. The probability of a connection between any two neurons in a block of tissue depends crucially on their distance. There are two ways of calculating this for neighboring pyramidal cells. One is based on the assumption that the connection via straight axon collaterals and that the form factor on the receiving side is described by the cross-section of the dendritic tree. So you can describe the shape through these metrics that he's collecting from the neuroanatomy and then model these as appropriate. So, what are the propositions which can be extracted from these anatomical facts? 4 of them are presented in Table 3. So we have the first one, which is A, and they involve certain measures, and that is most synapses are on spines. The second, which involves these other measures, most contacts are pyramidal to pyramidal. The third point is that the number of neurons afferent to a given pi cell is almost as great, around 5,000, as the number of its afferent synapses. Similarly for efferent synapses. So afferent are the ones heading towards a cell, efferent are the ones coming from a cell. And it's just a way, in this case, it's a neuroanatomical way to describe feed forward and feedback. Then D, which has another set of measures still. The cortex is a mixing machine. The activity is relayed in ever new combinations from one set of neurons to the next. So you have this idea, or this idea that was prevalent, I guess, early on, that the cortex is this all-to-all connectivity machine. But then, we kind of thought, well, maybe not, because there's, distinct function. And so, there's this idea of modularity. There's also this idea of a mixing machine, where there are new combinations generated all the time. And so, this kind of, I guess, is maybe the inspiration for connectionism, but it gives us this sort of model of, connections versus function. The upshot is proposition D. The cortex is a system of fairly uniform units, pyramidal cells, with a smaller number of neurons for different kinds of interspersed, connected to each other by widespread but very weak links. So weak, in fact, that the activity of one single cell can hardly exert an appreciable influence on the others. We must assume that the elementary event is constituted by fairly large sets of active pyramidal cells, so that fresh sets will be activated in succession through the synapses preformed in the cortical tissue and molded by a learning process. The picture I have in mind is influenced by Hebb's theory of cell assemblies, by Abellis's synfire chains, by various developments in this work by Palm. So I guess in this part of his career, he was really kind of thinking very theoretically in terms of what is the neuroanatomy I'm looking at mean? Eventually, of course, he'd go on to write the vehicles book, where he put all this together in these toy models called vehicles, and kind of stating out some of these, cleaning up some of these observations and stating them as the vehicle models. So here's a wiring diagram. Here, illustration of a naive neural model explaining some of the effects described by Hubel and Wiesel, who did the cortical columns processing basic shapes in visual cortex. So in A here, the array of dots represents the sampling points in a portion of the visual field corresponding to individual cones of the retina. So you have this visual field, you have the cones, they're dots. They're not like ommatidia, like we saw with the insects, but they do have this sort of spatial array that they're located in. And they represent different parts of the visual field. So this kind of shows how they phase shift, and then these two in C, it shows the connectivity between these different parts of the visual field as you rotate them 18 degrees. So these are things that belong to different orientation columns. So one orientation column represents the visual field like this, and the other one represents it like this. They're shifted in orientation in angle, but they represent the same spatial information. Then he has some nice drawings of orientation specificity. This is an array of hypercolumns seen from the top. So he's really into drawings and linking that with these histological images.